Good afternoon, children. We were learning the chapter transpiration. I am continuing the lesson to complete the portion. At the same time, after teaching the class, I shall send some revision question. You look on the textbook and write the answers, and if it is possible, send me back the photos of the answers you wrote in your rough book or on the paper. Then here let us see the kinds of transpiration. The transpiration from the aerial part of a plant occur from three different regions. We learnt through three regions transpiration occur. One is stomatal transpiration through the stomata. You know where the stomata occur on the epidermal part of the leaf. Usually on under surface of the leaves more stomata may be present. Sometimes in the upper surface also present, but in the lower portion you can see the stomata here. Stoma, the water vapor is coming out. That opening is known as vertical section of a plant part leaf showing diffusion of water vapor from its high concentration. You can see the stoma. The black colored two guard cells you can see, that are the kidney shaped cells of the stomata. That black colored cells are the guard cells of the stomata. You can see the minute opening, that stoma. You can see two guard cells are there, and surrounding the epidermal cells are also there. This is the diagram that may be asked to write. Draw. You will be asked. In many these kind of simple diagrams are usually asked by the board to draw. So it is a very simple diagram, but you should learn it properly. The stoma, the opening is known as stoma. Kidney shaped cells or bean shaped cell on either side of it is the guard cell, and the surrounding yellow colored cells are the epidermal cells. These three parts must be labeled by you. If the labeling is not there, you will not get the full mark. So labeling is important. Then certain other labelings also you can see there, that are chloroplast present in the guard cell. You can notice one thing, that only the guard cell in the epidermal cell, only the guard cells have the chloroplast. The surrounding epidermal cell does not have any chlorophyll, and you can see the nucleus of that kidney shaped cell or guard cell. These are the structures. The most important labelings are stoma, guard cell and epidermal cell. At least three labeling must be essential for a proper diagram to get the full mark. Then transpiration, stomatal. You know what is stomata. Then cuticular transpiration. Cuticle meaning that is a wax-like substance that is covering the usually on the dorsal side, the visible side, the upper side will have more a cuticle, a wax-like substance. Through that substance you might have observed the leaf, its surface is very smooth, and a glazing you can see on the upper side of the leaf. That is the dorsal side, that is covered by a wax-like substance, that is cuticle. Through that cuticle also transpiration or water vapor may escape, that is cuticular transpiration. Then lenticular transpiration. Lenticels are that are minute opening present on the woody stem. In woody big trees, the woods have small pores, minute opening on its stem, that is lenticels. Through three areas transpiration occur: stomatal, cuticular, lenticular transpiration. In this, the stomatal transpiration is more. The second place come to the lenticular transpiration. Only a small amount of water is escaped through the cuticle. So more amount of water, the most amount of water is transpired through the stomata. The second position come to the lenticular. The third position only come to the stomatal transpiration. Then let us see the mechanism of stomatal transpiration. You know that the stomata is usually, here you can see the section of a vertical section of a leaf. You can see. In that vertical section, the upper epidermis, it has an upper epidermis and lower epidermis. In between, mesophyll cells are there. You can see closely arranged mesophyll cell. That cells are known as palisade mesophyll. And in the lower side, on the lower part, you can see spongy mesophyll. Spongy mesophyll, spongy meaning there may be small openings or pores in between the cell. So it is known as spongy that have intercellular space in between the cell. So in lower part spongy mesophyll cells are there, and in the upper part the palisade mesophyll cells are there. In the cross section you can see. Then again you can see a very large intercellular space is there. That space is known as substomatal space. Here you can see the water vapor is diffused out from the surface of the cell. You can see very high concentrated presence of water vapor is present in the substomatal space. Above the stomata you can see. Then the water vapor may be diffused out through the pore known as stoma. Again you can see another thing, that usually the stoma may get opened when this guard cell, kidney shaped cells, you can see this guard cell become turgid by absorbing water. When water is filled with this kidney shaped guard cell, that cell become very turgid. Here one structural peculiarity is there for this guard cell. Its outer, it has two walls are there. It has an inner concave wall and a convex wall on the upper or the outer area. The outer area is stretchable so that the inner, that thicker wall will not stretch that much. So when the cells become turgid, the opening will become wider because of its peculiar variations in their cell wall. That stoma will become wide when that kidney shaped cells become turgid, so that the openings wider, so that more water vapor may diffused out. You can see when the stoma become wider, the substomatal space have a lot of water vapor that may escape out through the process of diffusion. You know diffusion, what it is. From higher concentrated area, molecules have a tendency to spread to the lower concentrated area. That is how the diffusion of water vapor will occur. Then again in the next picture you can see the movement of water molecule. Here certain arrows you can see. The xylem vessels in this section of this leaf, you can see the xylem vessel. You know that from the roots, when plant absorb water, that pass through the xylem vessel to the upper part of the plant, and finally it reach the leaves. You know that leaves have vascular bundle. xylem and phloem together known as the vascular bundle. Finally, the water reached the xylem vessel. From the xylem vessels, you can see that the arrow shows the movement of water molecule. The water molecule may be transferred from one cell to another by osmosis and by the process of imbibition. The water molecules may travel through the cell wall of these cells. Finally, it reaches the intercellular spaces. There you can see the water vapor that may be evaporated, and you can see the moment of evaporation. So that substomatal space may fill up with water. So it may diffused out. So the arrows, certain xylem vessels may be marked as A, and then the substomatal space may be marked as B. In question like that, the section may be there to mark the part. You must label the name parts labeled A, B, etc. What does it show? That arrows shows the movement of what, or the arrows is represented by what. Such questions may be there. It represents the movement of water molecule. Such questions may be there after showing the diagram. This will not be asked to draw, but the stomata structure may be asked to draw for the board examination. This may be asked like that. By after showing the diagram, there may be several questions. Stoma, everything you must learn the labeling portion and to identify each structure. Only labeling or question may be asked after showing the diagram. You know that. Then again, another experiment is given here. Such a leaf is known as a dorsiventral leaf. Here, an upper surface and a different upper and lower surface of the leaf may be different by its structure. Such a leaf is known as dorsiventral leaf. That on either side of the leaf, a cobalt chloride paper is attached. You know what is called cobalt chloride paper. In the last class we learned. It is an indicator of moisture. Again, here you can see on the upper and lower surface cobalt chloride paper and is attached. That is attached to the leaf by using the glass slides. You can see two glass slides are present by covering that cobalt chloride paper and is held in position using the clip or by putting a rubber band that can keep it in position. Then after 1 or 2 hours, if we check the cobalt chloride paper, the paper on the lower surface of the leaf will have changed its color very fast. That lower surface, the cobalt chloride paper change its color, blue change into pink, indicate the presence of, that will change its color very quickly. It shows that more evaporation take place through the lower surface of the leaf. That is, more stomata is present on the lower surface of the leaf.